Hello everyone, welcome to today's live broadcast, Development of Selection Strategies for Homologous Recombination Deficient HRD Patients Utilizing a Liquid Biopsy. I'm Robert Castellanos of Labroots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots and sponsored by Epic Sciences. Epic Sciences, Inc. is a privately held diagnostics company that is developing highly sensitive tests to identify and molecularly characterize circulating tumor cells, CTCs, through a minimally invasive blood sample. We've developed a powerful, rare cell detection and characterization platform, which was founded on unique technology, exclusively licensed from the Scripps Research Institute. This unbiased and enrichment-free approach will empower the personalization and monitoring of cancer treatments. Our partners include more than 37 pharmaceutical and biotechnology companies, major cancer centers, the National Cancer Institute, the Institute of Health. These collaborations use Epic Sciences' platform to observationally monitor changes in the composition of CTCs during the course of study. Furthermore, our no cell left behind technology can help match patients to the right therapies and monitor for drug resistance so that the best treatment path can be chosen at every clinical decision point. For more information, please visit epicsciences.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the C button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would like to introduce today's speaker, Mark Landers. Mark Landers has 20 years of experience developing and commercializing molecular assays to drive translational and clinical research. In his current position as VP of Translational Research at Epic Sciences, he is leading the development of assays to characterize CTCs by genotype and phenotype at the single-cell level. These assays can be utilized to develop clinical tests to drive patient care decisions. Prior to his tenure at Epic Sciences, Mark was head of R&D at Althea DX, where he's developed several campaign assays for targeted therapies. He has also held several senior-level scientist positions in the San Diego biotech community at companies including Life Technologies, Sierra Gene, and Bical. I'll now turn it over to Mark Landers for his presentation.
Thanks, Robert, and thanks to everybody who logged in this morning for the talk. So the focus of the talk is on detection of HRD and CTCs from metastatic castrate-resistant prostate cancer patients using the Epic liquid biopsy. But before we get started into detection of HRD, I'll give a little background on the Epic CTC platform, how it works, why it's different, and then focus on how we've leveraged that platform to develop predictive tests that really can answer key clinical questions at clinical decision points that oncologists face. Then we'll focus on detection of HRD and CTCs and how we can see HRD in CTCs, then how we've used a phenotypic signature to predict HRD genomics, and then finally how we can use that phenotypic signature to predict HRD as well as predict response to PARP inhibitors. And so first, a little background on Epic Sciences. So I think there was some mentioned in the intro. In 2008, we were spun out of the Scripps Research Institute, and we're currently located in a 40,000 square foot facility in San Diego that's shown in the lower left-hand panel. We are CAP accredited, CLIA certified, and currently support over 90 clinical trials. This is with our 40 or so pharma partners, or 25 academic partners. We've developed capabilities that allow us to run our CTC assay, but we also have integrated capabilities, which we'll describe further, that include DNA FISH, qPCR, and single-cell NGS. And so we'll dive a little bit into the capabilities as we go through the background. And so I'm going to focus a little bit on the Epic platform and how it works. And so traditionally, since the discovery of CTCs in the 1800s, there's been a lot of ways to try to isolate CTCs from the blood. And so as people have developed different approaches, they've either targeted surface markers like EpCAM, or they've tried to use the differences in size between the tumor cells and the white blood cells to filter out CTCs or changes in density. All of these approaches, though, require that you know something about what a tumor cell looks like or is. And so the key difference with the Epic platform is that we are taking all nucleated cells from a blood draw, no enrichment, no selection. So this really allows us to see a much wider biological range of CTCs from a single blood draw. And so I'll walk through how it works. So at a clinical site, a simple peripheral blood draw. This is taken, it's a 10 mil tube, so we get about 8 mils of blood in there. It's room temperature shipment to one of 3 sites. So we have in North America, we ask that you ship the samples to our site in San Diego. For European trials, we have a relationship with LabCorp, which allows to ship samples to Belgium. And then we also have a site in Singapore, which can handle Asia-Pacific trials. Basically, once the blood is drawn, samples are received. Within 72 hours of blood draw, we like to process the samples. We can process them up to 96 hours, but preferably best performance is 72 hours post-blood draw. So once the sample's received, red blood cells are lysed, and all nucleated cells are spun down into a pellet. These cells are then resuspended and deposited onto the surface of glass slides. Typically, from a single tube, we make about 10 slides. And so these can then be either frozen away or assayed immediately. And so we have data showing that these are quite stable once frozen. So I think we have a stability study out to 2 years. And so typically 2 slides are assayed per assay, and that means that we create a little biobank from each sample, so those can be stored in the freezer, which is really useful for when you want to look at additional markers later or bridging studies. So the assay to look for CTCs is an IF staining assay, and so this consists of a cocktail of antibodies directed towards different cytokeratins that help us identify epithelial cells, an antibody against CD45, which helps light up the white blood cells, DAPI, which characterizes the nuclear staining, so it tells us which cells have intact nuclei, and then we have additional room for additional markers. And so in this case, the biomarker, and for most of the data we'll be showing you, will be AR, or an AR isoform. And so typically we also have assays for breast cancer that target ER or CMYC. There's a lot of things we can target there, and so we have a menu of test items, as well as doing custom assay development. So after the IF staining procedure, each slide is scanned, and so this takes about 5 minutes per channel, each slide. So we're scanning over 3 million cells per slide, collecting data on not only the IF fluorescence intensities, but also on cell morphology. And so really, then what does the heavy lifting here is the proprietary algorithm. And this is looking at the cells on the surface of the slide the way a pathologist would. So it includes not only the cytokeratin expression, CD45, but also looking at the morphological features of the cell. And so from that algorithm, we generate a list of candidate CTCs. Those CTCs are then confirmed by pathologist-trained staff, and then we have a list of CTCs and can enumerate and characterize them further. And so when I talked about a wider biological range of CTCs that we see, this is what we're talking about. And so the upper left panel is showing what the surface of the slide looks like. And so mostly what you're seeing there are white blood cells. So typically we can see, we know what white blood cells look like, and then we can look for rare events. And so what's shown below that are what we call classic or typical CTCs, and these are much larger than the surrounding white blood cells and have brighter cytokeratin expression. These are what we call classic CTCs. But what's more important than that, or what is important as well, are those shown on the right, where we see small CTCs that other platforms might miss. We can see apoptotic CTCs, where we see they no longer have an intact nuclei, but can characterize the expression of different biomarkers on the surface. EMT type cells, where we no longer have, they're typically epithelioplastic. The cytokeratins are not very bright or not expressed at all, but we know that these are still expressing our biomarker of interest or have morphology consistent with a CTC. We can also see CTCs in clusters, in any of these subclasses of CTCs in clusters. And so one of the questions we're commonly asked is, well, what does that mean, and why is it detecting more CTCs better? And so we did a head-to-head comparison, and this is work done in conjunction with Memorial Sloan Kettering, and this was a head-to-head comparison versus CellSearch. And so metastatic castrate-resistant prostate cancer patients were, a blood draw was taken prior to treatment with either abiraterone, enzalutamide, or taxane in a first-line, second-line, or third-line setting. A replicate sample was taken for CellSearch analysis. And then here all we're simply doing is comparing how many CTCs we saw at that particular blood draw. And so this is data from 173 patient samples. And so the top panel is showing the CTCs per mil detected by Epic, and that's shown in the blue bars. The red bars show the CTCs detected by the CellSearch platform. Right below that is the difference. And so you can see typically where you see all the blue bars, that's where Epic detected more CTCs than the CellSearch platform. And even more critical to that is the clinical sensitivity. And so how many patients did we see with CTCs where we could then go on and characterize different biomarkers on those CTCs? And if you look at the table on the bottom, from the 173 patients that we looked at, with CellSearch, CTCs were present in 67% across all lines of therapy, whereas with the Epic platform, we detected CTCs in over 96% of the patients tested. So much better clinical sensitivity. And then when you ask the question of why are these CTC subtypes that other platforms are missing, are they important? What we did is we looked at, okay, if a patient had a prevalence of these CTC subtypes, what did that mean? How did those patients do in comparison to other patients? Were they biologically meaningful? And so shown on top are patients who had a prevalence of cytokeratin negative or epithelioplastic CTCs, and how they did in comparison to other patients within the cohort. And so that's shown on the red line of that Kaplan-Meier curve. And then the significance of that is shown over on the right in the multivariate model, where you see the red p-value. And so it was significant that these patients did much worse if they had a prevalence of CK negative CTCs. And even more drastic was the prevalence of small CTCs. And so you can see a dramatic difference in overall survival. So these different subtypes lead to much worse prognosis for these patients. And so beyond enumeration, we've done a lot of work to integrate in downstream technologies that can really help us characterize the CTCs at a much more detailed level. And that's really will be some of the tools we use today when we talk about how we've leveraged the platform. We use a lot of these tools. And so beyond just identifying the CTCs and enumerating them, we have a variety of protein markers which are very useful. But beyond just expression of those different proteins, we can also characterize the localization of it. And as I'll show you later, nuclear localization of particular, like the ARV7 marker, is critical to defining the clinical specificity. We've also integrated in capabilities like DNA FISH, where we can remove the cover slip, add FISH probes after staining, and now we're integrating genotype and phenotype information. As part of the process, we're also collecting XY coordinates from each cell, and this allows us to pick single cells off the surface of the slide and then do downstream qPCR and next-gen sequencing, and we'll show you some data from that today. And so now I'm going to focus a little bit more, less on the platform, but more on how we've developed predictive tests leveraging this platform. And so we want to develop tests that answer specific clinical questions that an oncologist might face. And so one of the first questions we sought to answer was, in metastatic castrate-resistant prostate cancer, patients often get abiraterone, enzalutamide, or AR-targeted therapies, or they're put on taxanes or cytotoxic chemotherapies. The question is, which patients would respond better to which therapy and when to give which therapy? And so work we did that was published earlier this year, and this is work with MSK and Howard Shear's group, and so the interesting part here is that this is targeting ARV7. So the ARV7 is a truncated version of the androgen receptor. It's missing ligand binding domain. It's constitutively active, and it's been shown that expression of ARV7 has been associated with resistance to AR-targeted therapy. And so we looked at, of the patients we looked at here, and so this is showing you patients with a prevalence of ARV7 expressed and localized to the nucleus showed, when given AR-targeted therapies, had very good specificity and was only detected in patients who had PSA progression. And that's shown in the upper left panel, where all the dots that you see that are where we detected ARV7, those were all in patients who had PSA progression when given an AR-targeted therapy. More dramatic than that is if you look over on the far right panel, is when you gave these patients AR-targeted therapy, and those are the ARV7 positive patients are shown on the red line of these KM curves. These patients did much worse. They had a hazard ratio of 11.45 when given the AR-targeted therapy. So clearly not a good choice of therapy for these patients. But what we wanted to understand better is how much better these patients did when you gave them a taxane chemotherapy. And so shown below is the therapy interaction that we were able to demonstrate. And so when these patients who are ARV7 positive were given a taxane instead of given an AR-targeted therapy, we showed that their hazard ratio was reduced to 0.24. And so this is a 76% reduction in risk for these patients. So this is a very good predictive test and can be used in a first or second line, third line setting to identify patients who should not be given abiraterone, enzalutamide, or other AR-targeted therapies and should be switched to taxane chemotherapies. And so in the same clinical setting, we really wanted to understand, because as we looked at these patient samples, we saw a wide variety of CTC subtypes. It's really, in metastatic castrate-resistant prostate cancer, it's a lot like the Wild West. You see a lot of different crazy cell types in these patients. So what we really wanted to understand is, when is disease heterogeneity so high that no targeted therapy will work and you should really be using the taxane chemotherapy? And so we looked at 221 patient samples, about over 9,000 single CTCs from these patients, and we sought to answer the question of what is high heterogeneity and what's its effect on being treated with either the targeted therapy or chemotherapy. And so the way we did this was following the typical scanning procedure. We did high content image analysis, and so basically this is doing a lot of segmentation of the cells, the nuclei of cells, and then we're able to measure a lot of digital pathology features. And so the things we can measure, in addition to the IF expression at different markers, were nuclear area, nuclear to cytoplasmic ratios, circularity, about 30 different measurements in total. And so we're doing this for each cell on the surface of the slide. From those features, we then use principal component analysis to mathematically come up with a model for how many of these features clustered together and made unique cell types. And so based on that analysis, we were able to come up with 15 different CTC phenotypes that were present across the entire cohort. And so what was interesting there, and what's shown on the left, is the prevalence of these CTC subtypes labeled A through O across the entire cohort. And so this is in bulk looking at how the prevalence of each CTC subtype. But what's more interesting is when you look at the patient level. And so the heat map shown here is starting from the left to right. Patients, each column represents a patient. Each row represents a different subtype. Where you see color, and the darker the red, that's the higher prevalence of that particular CTC subtype. And so the patients with more CTC subtypes, it's correlated to bigger diversity. And so as you see, as we move from first line to fourth line, you see the prevalence of multiple subtypes within patients got higher. And so we wanted to measure this, and so we applied a measurement called the Shannon Index, and this is a diversity measure that's commonly used by ecologists when they go to a new environment and they want to characterize the diversity that they see within like a rainforest. They would count up the different species that are present. And then based on that, you can score the increase in diversity. And so that's what we applied here, treating the cell types as different species. And so when you look at the bar chart on the right, you can see that the Shannon Index, as diversity increases, the number increases all the way up to over 2. And so in the first line, we saw heterogeneity, but we did see heterogeneity increase as you move from first to fourth line. But what was particularly interesting to us was that we saw very high heterogeneity in first line patients, and we also saw very low heterogeneity in fourth line patients. And so it became really apparent to us that these different CTC subtypes could be very critical to characterize further. Before I get to that, I'll show you what the effect of the different heterogeneity score is. And so we took the top 25% of the patients by Shannon Index, so the patients with the highest heterogeneity, and looked how well they did when they were given AR-targeted therapy or when they were given taxane chemotherapy. And so when you look at the KM curves on the top, that's showing on the left progression-free survival, and on right the overall survival of these patients. And you can see that when these patients with very heterogeneous disease were given AR-targeted therapies, they had a hazard ratio of 5.5. However, when they were given the taxane chemotherapy, they had a hazard ratio of 1.6. So they did much better on taxanes in comparison to the AR-targeted therapy. And so again, we look to show the therapy interaction, that these patients with high heterogeneity, when given the taxane chemotherapies, had a reduction in risk. And so here we saw their hazard ratios. When given the taxane chemotherapy, their hazard ratios were reduced to 0.323. And so there's a significant reduction in risk when you take these patients with high disease heterogeneity and give them a cytotoxic chemotherapy versus AR-targeted therapy. And so beyond that, as I said, we were characterizing all these different CTCs and CTC subtypes. We were able to incorporate our single-cell sequencing to look for the clonality of these, look for different drivers that were driving different subclones, and look how those subclones did across the course of treatment. And so this is showing CTCs from a single patient. And so you can see there were 8 CTCs detected in this patient that had very flat genomes. What's shown here are whole genome copy numbers. Each dot represents a 1 million base pair window in the genome. And where you see the dot above the 0 bar, that's an amplification. Where you see it below, that's a deletion. And what was interesting is that you can see different clonal species with different genomics. And so you can see the 5 patients on the middle bottom right there who had AR amplification, but there were also patients that had unstable genomes where we actually saw that they didn't have an AR amplification. So we actually saw a branching event there. And the reason this is important is we actually found particular CTC subtypes that didn't do well on either type therapy. And so this is important to what we're going to talk about today, but this cell type K, and this was presented by Howard Scher at ASCO GU earlier this year, there was a particular CTC subtype. This had large nucleus, high nuclear entropy, the presence of nucleoli, and when you had a prevalence of these type of cells, these patients did particularly poor on AR-targeted therapies, but also didn't do very well on the taxane chemotherapies. When we picked and sequenced these cells, we saw that they had a high prevalence of genomic scarring, consistent with what you'd find in HRD. And so these patients had a higher prevalence of large-scale transitions, and we'll get into this a little further. But this really was what made us think that we could particularly characterize HRD using morphological measures. And so I think that this is where we're going to dive a little bit into HRD and PARP inhibitors and how HRD is predictive of PARP inhibitors. So after setting the stage, I think I'll take a step back and characterize and provide background information on how PARP inhibitors work, how people are out there measuring HRD, and then how this test is different and what the advantages and disadvantages are. And so the mechanism of action for PARP inhibitors basically target the single-strand repair pathway. And so PARP inhibitors are targeting PARP. PARP can no longer participate in the base excision repair pathway. You accumulate a lot of single-strand breaks, and you're unable to fix those, which then lead to double-strand breaks. In a normal cell, the accumulation of double-strand breaks is repaired by the homologous recombination pathway with very high fidelity. In patients who have a mutation in this pathway are unable to fix their DNA, you get double-strand DNA repair by the non-homologous end-joining pathway, which is much lower fidelity and leads to much more unstable genomes. And so PARP inhibitors are particularly effective in the patients that have homologous recombination deficiency. The big question is how do you find these patients? And so traditionally, people have looked at a variety of genomic markers, and so there's been a lot of work published showing that BRCA mutations in the germline in ovarian are often associated with HRD. And when you expand that pathway out into to including ATM, PALB2, other pathway members, and look in look for somatic events in tissue biopsy, that number increases to roughly 20%. And then others have then looked for marks of genomic scarring, and I think we showed some of this earlier, but they look for large-scale transitions, allelic imbalance, or loss of heterozygosity. And the advantage to this is you're actually looking for the effect of HR deficiency on the DNA. So the caveats to all the tests on this board are they're genomic-based tests, so it requires interpretation of the variants. You have to understand the impact of the variant, any allelic frequency cutoffs when you're looking in tissue. And then you also have to be able to set cutoffs and interpret these. They're all dependent on, in the somatic setting at least, on biopsy, and we think that there's considerable issues with biopsy in the metastatic setting. And then all of these, because they're sequencing-based tests, have much longer turnaround times. And so thinking about what's out there and the current methods that have been used to characterize HRD, our thought process was that we wanted to develop a test that could be run from a simple blood draw that would provide single-cell resolution that could allow us to better understand the heterogeneity that's present in a patient, but could also detect somatic HRD, so sub-clonal HRD, patients that have a subclone that has HRD, but maybe not the entirely germline disease state. We wanted to develop a test that was non-sequencing based, that could be performed in a week, had lower COGS, and equivalent or better performance to the current methods that are out there. But more importantly, and I'll show you the data from what we've generated today, is we wanted to be able to answer a specific clinical question in direct treatment. And so I think that's what we're going to focus on today, is how we've applied this to metastatic castrate-resistant prostate cancer. And so as we move along, that'll be the data that we focus on. And so we started out by saying, we understand how tissue sequencing is done and the markers. There's been a lot of publications showing tissue sequencing. So the first question we wanted to address is, could we see genomic markers of HRD in our CTCs? And so this is really data where we picked and sequenced CTCs. And what's shown here are whole genome copy number plots of individual CTCs. And so again, I'll walk through the process where post-staining, we remove the cover slip, we add some buffer to help us recover the cells from the surface of the slide, and then using a micromanipulator type instrument, it drives to the exact XY coordinates, recovers the cell from the surface of the slide, and transfers it to a single well of a 96-well plate. The cells are lysed, whole genome amplified, and then sequenced. And the data we're showing here right now is from low-pass whole genome sequencing. Then we can generate whole genome copy number profiles similar to what you would see in an array CGH experiment. And then we can look for not only focal amplifications and deletions, but the presence of large-scale transitions, so areas of greater than 10 megabase that are either amplified or deleted. This scores as 1 LST. And so what's shown here, when we characterize the number of LSTs per CTC, you can see an example of that on top, where the single CTC from a BRCA mutant patient had 45 LST, and a single CTC from a BRCA wild-type patient had very few LST. So we went back to some of the classic models for PARP sensitivity, and so there's been a lot of work published that has characterized genomic scarring in triple-negative breast cancer and ovarian. And so we took some triple-negative breast cancer cell lines, and then we spiked them into the system. They were spiked into healthy donor blood. We picked and sequenced these cells, and we looked for the presence of genomic scars. And so the two cell lines that are shown on the outside columns, the HCC 1365, as well as MB 231 to be our control, and MB 436. So the two on the outside, MB 436 and HCC 1365, these are our PARP sensitive BRCA mutant cell lines, and then the one in the middle is our triple-negative breast cancer cell line, which we know has a high degree of genomic instability, but is PARP insensitive. And so the idea here is that we would measure genomic scarring in the single cell level, and then we would see how can we best separate typical genomic instability that we would expect to see from HRD levels of genomic instability. And so what's shown on the far left are the number of LSDs detected in single cells, shown in blue, versus a control sample, which is just bulk DNA from that same cell line. And so you can see first that we can recapitulate the number of large-scale transitions we see per single CTC very well in comparison to the bulk control. But when we compared that to the number of LOH breakpoints we got by doing whole exome sequencing from single cells, as well as the cell line controls, we saw that the measurement of LSTs really separate these to high genomic instability PARP insensitive from high genomic instability PARP sensitive. And so this is what we used moving forward to train our phenotypic classifier for HRD. And a lot of questions are always asked about how this relates back to tissue, BRCA status. And so we sequenced a lot of patient samples here, and what this is showing is each column represents CTCs from a single patient, and we're characterizing the number of LSTs per patient. And the far left panel shows the patients in which we had a matched tissue biopsy that had a BRCA/ATM mutation, and the panel right in the middle shows the patients in which we had a BRCA/ATM wild-type tissue biopsy. And so what's interesting here is you can see that there's a much higher prevalence of LSTs in the patients in which there was BRCA or ATM mutations present in the tissue biopsy. But we also see HRD in the wild-type patients in very sub-clonal populations. And so this is the far right panel showing patients in which we were unable to get coverage of the entire region, so we couldn't make a wild-type or mutant status because there were holes in the coverage, or we detected mutations that we didn't know the significance of. And so clearly you can see high degrees of HRD in these patients in which we were unable to select by tissue sequencing. And so once we were pretty confident that we were able to see CTC or HRD genomics in our CTCs, we wanted to train up our phenotypic classifier that could then be used to predict that level of HRD genomics. And so to do that, we followed the same methods that we used when we were looking at heterogeneity. And so starting with patients, single CTCs from patients where we knew in their tissue biopsy they had BRCA or ATM mutations, we picked and sequenced the CTCs from those patients and characterized the number of LSTs. And then we did the high content image analysis for each of those cells. And so the idea here is that we're going to be able to compare genotype to phenotype at the single cell level in different patient populations, either a BRCA mutant, non-BRCA mutant, or wild-type patients. And so when we did this, we collected all of our phenotypic features, and we saw some of the phenotypic features and that correlated very strongly to the presence of genomic scarring, specifically of LSTs. And shown on the left is a panel showing some of these genomic features that we measure and their correlation to an increasing number of LSTs. And then when you look over on the panel on the right, these are showing the features ranked from top to bottom by either positive correlation or negative correlation to the presence of scarring. So then we can train up a model that using these features and do a lot of machine learning that will allow us to look at phenotypes and predict genotypes. And so just taking a 40,000 foot aerial view of what this looks like. So these are, when we do that and we create a model that predicts using just phenotypic measures to predict HRD, and then we go in and we're looking at the overall genomic instability that we see in those cells. So moving from left to right on this heat map are higher phenotypic HRD scores to lower phenotypic HRD scores. What's shown on each row are particular genes and the copy number variations we saw on those genes, red being amplified, green being deleted. And you can just see from a high level view on the far left where we saw and predicted high HRD using the phenotype, we also saw a much higher prevalence of copy number variation. And as we moved to the right, we saw a much lower prevalence of copy number variation in those CTCs. But what do those cells look like? And so this is really interesting when those cells have a particular phenotype. And the panel on the left shows a lot of images from HRD positive CTCs that had so high prevalence when we picked them in sequence. They had a lot of LSTs. And some of the common features shown here are that they had low eccentricity, high nuclear to cytoplasmic ratios, and a high nuclear texture. And so this is equivalent to what you'd see in like a black and white television, where we saw that there wasn't a very uniform, even DAPI staining procedure in comparison to the cells that were predicted to have low genomic instability, in which there was much higher eccentricity, lower or normal nuclear to cytoplasmic ratios, and normal nuclear texture. And so then we moved on to train and test the classifier. And so to do that, we took close to 600 individual cells across 35 patients And we picked and sequenced each cell individually and looked for the presence of LSTs, and that's what's referred to here as ALST. And we compared that to doing the high content image analysis for each of those cells and using the phenotypic measures to predict if it would have high or low LSTs. And so what's shown here is in the 2x2 matrix is where we predicted that it would have high LSTs by phenotype. We saw a high prevalence that it did in fact have high levels of genomic scarring. And so the accuracy at the single cell level was 78%. We saw a very good specificity. We had 86% specificity. And where it got even better was when we said, okay, at a patient level, if we required there to be 3 or more CTCs with this particular phenotype, how well did that match up to there being a prevalence of 3 or more CTCs with high genomic instability as measured by sequencing? And so the median of these values is shown below in the waterfall plot, where we had high levels of measured sequenced genomic scarring versus the predicted genomic scarring that we saw within an entire patient, and that's shown in blue. And so at the patient level, the classifier gets even more specific and more accurate. And so I think we've walked through how we do the CTC phenotypic characterization, how that correlates to HRD genomics, how those CTC genomics correlate back to HRD mutations in the biopsy sample. But really more importantly, we wanted to better understand is what does the prevalence of these HRD positive CTCs mean when you're looking at patients, and does it predict response to therapy? And so one of the first questions we asked is okay, well, what's the prevalence of this marker across cohorts of mCRPC, and what does it mean when those patients are given the standard of care therapies? Would they be better on a PARP inhibitor? And so we took cells, this is looking at 6,000 CTCs from close to 200 patient samples, where now we've trained the classifier, now we've only done the phenotypic analysis here. And we saw when patients that were positive for the HRD CTCs by phenotype, we saw in the first line 21%, in the second line 24%, and up to 40% in the third line of therapy. Across all lines of therapy, about 30% prevalence of the CTC-based marker using just imaging. What was interesting to us is we really wanted to understand how did these patients do when you gave them the AR-targeted therapy in comparison to the taxane, or in comparison to potentially a PARP inhibitor. So when given the standard of care treatment options, these patients did much worse. And so you can see the patients on the red line of this KM curve were given the AR-targeted therapy. Those are the HRD positive patients, and they had a hazard ratio of close to 7.7 when given the AR-targeted therapy. And when given, they did slightly better on taxanes, but still did worse than patients without HRD. And so these were similar to what we saw with our cell type K, and these might be the patients that you'd want to give the PARP inhibitor to. And so to investigate this further, working with Felix Fang and the team running the NCI 9012 trial, we collected samples from a trial that included a PARP inhibitor. And so to give you the trial design here, patients, these are first-line metastatic castrate-resistant prostate cancer patients prior to treatment with either abiraterone or abiraterone plus velaparib, the AbbVie PARP inhibitor. We collected a baseline sample, and then we collected a non-therapy sample. We put these samples on slides, and then we analyzed these slides for the presence of HRD positive CTCs, and then we looked to see if you had a prevalence of these CTCs, could we predict your response to PARP inhibitors? And so what's important to understand when you look at this trial, in the first-line setting, the AR patients had virtually no difference or no benefit in comparison to patients given the PARP inhibitor. And so you can see by the KM curves here, there wasn't any real benefit in mixing in the PARP inhibitor unless you used a selection device. And so I'll dive deeper into the response here, but what's interesting is we saw the prevalence of the biomarker in 30%, roughly 30% of patients that we looked at. And so this matches up very well with the other cohorts that we've seen from MSK, and now has been confirmed in multiple additional cohorts as well. We're seeing the prevalence in metastatic castrate-resistant patients across all lines of therapy at about 30%. But what was impressive to us is if you look at these waterfall plots shown in the bottom left, these are showing the max PSA decrease that we saw in each patient sample. Where you see red bars, you're seeing our biomarker positive patients, and where you see blue bars, you're seeing our biomarker negative patients. And what's important to consider here is where we didn't see CTCs, those patients were scored as biomarker negative. We only counted a patient as biomarker positive if they had CTCs present and they had our HRD phenotype. And so what's shown in what's interesting is that in the A arm where we treated with only olaparib, our biomarker positive patients showed a PSA response. Only 22% of them responded. When you look in our B arm, where we actually, if we were to use our biomarker positive patients, 93% of our biomarker positive patients showed a PSA, max PSA response. And then across in the biomarker negative patients did about the same across both arms. But what's interesting, and then when we looked over at the overall survival of these patients, we saw that biomarker positive patients did better. And so when you look on the KM curve shown on the left, this is with no selection device. We saw about equal response of patients whether they were given the PARP inhibitor or not. When you look at with selecting patients with our CTC classifier, you can see those KM curves start to separate. And so this is very encouraging to us. I think with more N, we'll actually see that get to statistical significance fairly quickly. But what we also wanted to do is confirm that we were actually targeting the cells we thought we were. And so we looked at, using our longitudinal draws, we actually looked at the prevalence of HRD positive CTCs from the change in the prevalence of those CTCs from baseline to on therapy. And so when we look at the A arm, you can see these patients were given only abiraterone. When there was a prevalence of HRD positive CTCs at baseline, we saw 166% increase in HRD positive CTCs on therapy. In contrast, when you look at patients in the B arm, there were we saw a dramatic decrease in HRD positive CTCs after therapy. So we were targeting the cells we thought we were. And so we saw 82% reduction in the prevalence of HRD positive CTCs when you included the PARP inhibitor. And so that's another validation that tells us, yes, we can predict response to PARP inhibitors, and then we can look longitudinally to say, yes, in fact, we are getting the cells we thought we were getting. And so I'll wrap up at this point, and just some summary ideas is that, because of the wider biological range of CTCs that we're able to detect, the CK minuses, small CTCs, and the integrated technologies that we use to better characterize these CTCs, both genomically as well as phenotypically, we can develop predictive tests that answer key clinical questions such as the ARV7, heterogeneity, and now response to PARP inhibitors. Heterogeneity has a huge impact on these tests, and so I think single cell resolution is going to be key to really identifying sub-clonal somatic tumor types and monitor their progression throughout treatment. And we were able to demonstrate that we could in fact do that with our HRD signature and in response to PARP inhibitors. And so I wanted to also say thanks to a lot of the work that was done here was in conjunction with collaborators. And so the team at MSK and Howard Shear's team in the NCI trial, and Felix Fang and Maha Hussain, and all those who helped us get those clinical samples. And then the Epic team here, who's really driven this from CTC to actually developing predictive tests that can be useful to direct treatment. And so I think we have 10 minutes left here, and I think this is where we'll open it up to questions. So I think back to you, Robert, for any of the questions.
Perfect. No, thank you so very much for your wonderful presentation. And before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We're going to try to answer as many questions as we possibly can, and all questions that are not answered will be followed up with at a later time. Question number 1. Currently, most PARP trials require screening for BRCA and ATM mutations. Can you comment on how this test fits into this landscape?
Yeah, and I think that was one of the, hopefully I highlighted that during the talk, but one of the key differentiators of this test is that we're not reliant on a fresh biopsy. And so when you think about metastatic castrate-resistant prostate cancer and the lack of being able to get a biopsy and the expense incurred in taking a biopsy, obviously the liquid biopsy has a lot of advantages. And in many cases, taking a biopsy it's unavailable or almost unethical to do to these patients in late-stage metastatic disease. The test is not sequencing based, so I think this is a key differentiator when you think about how we take this test and have a quicker turnaround time, as well as globally deploy this and its application as a diagnostic. This test has key advantages over sequencing in the fact that a lot of labs around the world can do an IF stain, and so simple blood draw, we can enable longitudinal monitoring, and so those are the key differentiating factors. So I see this fitting into the landscape that we're able to differentiate by addressing some of the complications associated with finding HRD in metastatic disease.
Thank you. How does the prevalence of CRPC HRD positive patient in your study compare to those of the TOPARP study?
Yeah, that's a great question. The TOPARP study was actually in third-line patients, mCRPC patients, and those patients were pre-screened for not only disease progression, but the prevalence of over or greater than 5 CTCs per mil. And as we showed earlier, the patients who tend to resist the AR-targeted therapies and chemotherapies, they tend to do, HRD positive patients tend to do worse on those therapies. And so it's possible that you're enriching for these patients in such a late-stage setting, whereas I think the data we were shown was much earlier in a first-line setting where these patients hadn't developed resistance mechanisms to AR-targeted therapies or taxanes. And I think that they had noticed or observed and reported 33% prevalence of HRD in this late stage. In third line, we saw the prevalence of the marker at about 40%, so those are very similar numbers. And then the key to this is that there were several patients that were excluded from TOPARP that were excluded because you couldn't get tumor biopsy or you didn't see CTCs. And so when you think about that, we're not excluding patients. If they didn't have CTCs, they were biomarker negative. And so I think this is some of the highlights, really, some of the advantages of a liquid biopsy, but also highlights some of the advantages of using a phenotypic analysis and comparison to genomic sequencing.
Thank you. It looks like we have time for one last question. What is the impact of heterogeneity on assay's accuracy?
Yeah, and I think this is really critical not only to this marker, but also just across all metastatic disease and late-stage disease. And so as we showed that there's a high degree of heterogeneity, and these patients do much worse on the targeted therapies. But when you think about it in the context of HRD, we saw sub-clonal populations of HRD present in a lot of the patients who showed some response. And so these patients, when you think about the undersampling that can happen with the tissue biopsy and other complications associated with there being multiple sub-clonal drivers and that effect on interpretation of the sequencing results, what you can see is that it's likely to have an impact in selecting these patients. And so I think the single cell resolution of this and the ability to look at the cells both phenotypically and genotypically at the single cell level is really what's driving the selection of these patients who maybe have subclonal disease and could show some response to the PARP inhibitor versus missing these patients due to sampling error or having the heterogeneity too high that they can't see the signal in the tumor biopsy. And so, I think that's. Any other questions or?
Well, no, just wanted to know just your final comments for today's presentation.
Yeah, and so we will address any additional questions. So please feel free to shoot additional questions in. We've presented this data, I think, at ESMO. We have it up on the website. We're happy to discuss further or dive deeper into this if there's an interest. And so I think as follow-up, if you go ahead and submit a question, we'll get back to you individually. And we also really look forward to the future path for this, and as we expand the applications of use. Obviously PARP inhibitors are a growing class of drugs. They're being used quite readily in new indications such as prostate cancer, squamous, non-small cell type indications. I think that's where getting a biopsy is very difficult and very heterogeneous disease has been observed. So I think it's very exciting how we can apply this marker going forward.
Well, thank you again, Mark Landers, for your awesome presentation. I'd also like to thank our sponsors, Epic Sciences, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through February of 2017. You'll receive an email from Labroots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Until next time, goodbye.